Well, I want to welcome all of you, and I want to welcome our guests from Bend, Cascadia College, and people from Bend K through 12. Are you there, Bend? Well, I can see them right here. Hopefully they're hearing it all. Okay, great. And I'm Mike Posner, and I'm the coordinator of the Brain Biology and Machine Initiative at the University of Oregon. Under the leadership of Richard Linton, the Vice President for Research here, and Bobby Haggerty over OHSU, we're trying to develop, and this is part of our effort, to develop a statewide work in brain and education. And OHSU and the U of O are cooperating in this venture. Tonight, we have with us a number of people from Bend, Oregon, who are going to also be able to ask questions thanks to the new technology of simulcasting. Can you all hear me in Bend? Yeah. Can you hear me? Yeah. No, you can't, I guess. Well, you can. Oh, okay, good. Yes. Okay, so when the lecture's over, then I'll call on people in Bend and also people from the audience here. Tomorrow morning, we'll also be joined by Portland and LeGrand. So we'll have somewhat of a statewide effort, or at least the beginning of a statewide effort. And in our Brain and Education series, last year we honored 2 books that came out in this field. One on Overcoming Dyslexia by Sally Shaywitz, and the other, Original Intelligence by David Premack. And we had a very successful day-and-a-half conference. This year we have 3 world leaders in the area of brain plasticity. And we're hopeful that this will provide a very strong impetus for our Brain and Education series. Tonight, Helen Neville, and tomorrow morning, Michael Merzenich and Elizabeth Belke. Helen Neville is currently the Robert and Beverly Lewis Endowed Professor at the University of Oregon and Director of the Brain Development Lab here. Helen is a native Northwesterner, educated in Vancouver, British Columbia through the master's degree, and then she went off to Cornell University where she received her PhD working in the neuropsychology of language. Her work at the Salk Institute using deaf children of deaf adults, that is native signers of American Sign Language, showed clear changes in the visual system as a result of auditory deprivation. That is, their visual system adapted to the lack of auditory input. These findings questioned the then prevalent view that sensory systems were hardwired prior to birth and provided impetus for the developments in neuroscience that's led to a strong emphasis on plasticity. Subsequently, after returning to the Northwest at the University of Oregon, Helen has worked on brain changes involved in learning sign using neuroimaging and also of spoken languages, and provided some of the best evidence for a variety of critical periods that might be of particular importance to educators interested in language learning. Her most recent exciting work concerns training of preschool children who might be at risk for failure in elementary school. Helen's pioneering work in brain plasticity has been recognized by the Lewis Endowed Chair by invited lectures all over the world, including in the Vatican and with the Dalai Lama in India. We are very proud that we have a world leader in brain plasticity here at Oregon, and it's very fitting that she starts this series of lectures under the title Experience Shapes Human Brain Development and Function. Helen.
Is this working? Can you hear me now? Can you hear me now? Huh? Yeah? No. How about now? Pretty good? How about now? Probably would help if I had it pointing up. Hi, Judy. Okay. Good. Okay. That was a great introduction. Thank you. And it's wonderful to see such a big turnout for a Friday evening after a long week, especially for you teachers. It's great. I'm really glad to have this chance to tell you something about the research that we've been doing in our lab on human brain development, and most particularly looking at the effects of input from the environment in shaping the development of the systems of the brain important for cognition and learning. What I'd like to do is first remind you a little bit about the fact that the human brain is so important. Tell you how fragile it is, which you may not know. Tell you a little bit, just remind you that it's highly differentiated in the adult, but then talk about the very long time course over which this differentiation occurs, and the effects of experience in the development of the systems of the brain important for vision, for hearing, for attention, and language. Then I will just say, well, yes, genes are important in brain development too. In fact, gene expression is very different depending on the environment children are in. And then talk about our work where we're trying to sort of sort out the contribution of genes and environmental factors in brain development and how we're doing that. And Mike heralded that a little bit, because the way we're doing it is trying to push things around by doing little interventions. And we're very happy to talk about that work tonight too. So here's a picture of a human brain from the side. This is the front, this is the back, like this. And it's always astonishing when I look at a real human brain of a once alive person. I didn't bring it tonight. That was close to the dinner hour and so forth. But you know, it's really small. Your brain is really small. It's about 2 to 3 pounds. It's about the size of your 2 fists put together. And it is really wrinkly, and it doesn't look very important, really. And it looks like, well, it could have been washed up on the beach or something. You know, it doesn't look very important, but, you know, then you just remember what you've heard and read 1,000 times, that the brain is important in interpreting all the information from the world that comes in our eyes, our ears, our noses, our fingers, and so forth. The eyes are only half of the equation. If you don't have this part of your brain, you're basically blind, because this part of the brain interprets the vision that comes in through your eyes. And that's true of every sensory system. Similarly, every move that we make in the world, every movement of our muscles is controlled by specific areas of the brain. Our ability to make music, walk, dance, speak, and so forth, everything is dependent on other parts of the brain. And of course, our cognition, all the mental activities, including cognition, like attention, our ability to make memories, our ability to store memories, to learn, to think, and language depends on other parts of the brain. And it is also true that every emotion that we experience, every aspect of social interaction that we experience is mediated by different and particular parts of this very undistinguished looking organ, which, as a matter of fact, is extremely fragile. So the brain in your head right now is the consistency of room temperature butter. I have a butter brain on my desk in my office. If you ever come and see me, you just poke it, just see how fragile that is. And of course it's encased in a hard bone, the skull. And indeed, that's a problem sometimes, because if a parent shakes a baby to get his attention, then you have a room temperature butter consistency tissue banging against a hard bone. These are just some important, simple facts that a lot of parents don't know. They don't know why you shouldn't shake your child to get his attention. And that is, of course, also why we wear helmets, because bones get broken and so forth. Well, what is this all-important fragile organ made of? The human brain is made of cells that are called neurons. Here's a schematic of one. This is the cell body. It has a long tail whereby it sends out information to other neurons. And on this sort of tree-like structure, it receives messages from other neurons. In fact, each neuron in your brain receives messages from about 10,000 other neurons. And there are about 200 billion neurons in your brain. And the way they communicate with each other is by generating electricity from chemicals. So there's electrochemical signals that are sent down here. And this is where this particular neuron is making a signal to another one. This is a blow-up of that contact. And these connections are called synapses. So inside your human brain right now, you have about 200 billion neurons. And here's a picture of one. This is a photograph of a real neuron. This is the cell body. This is the tree-like structure that gets messages from 10,000 other neurons. The tail isn't shown here. But since you have 200 billion neurons and each one gets 10,000 inputs, that means you have hundreds of trillions of connections in your brain. And that's how brains work, by specific and different weighting and unweighting of connections. That's how we learn. That's what a brain system is. It's a pattern of connectivity. Yes, it is complicated. That's why we have this multidisciplinary research consortium called the Brain, Biology, and Machine Initiative at the University of Oregon, where we take expertise from many different domains to work on the difficult problem of understanding the human brain, how it works, how it develops. Of course, you need good behavioral scientists, psychologists who know how to turn attention on and off, memory on and off, so we can image it in our imaging machines. We need good people who understand brain science, people who understand genetics and molecular biology, because that's important in shaping the brain as well. Chemistry, so we can know the electrochemistry of the brain. Physics, to get the beautiful images I'll show you. And computational science, to deal with the huge amounts of information that are generated with current neuroimaging techniques. Why do people around the world study the brain? Of course, we have several goals. Very clearly, this is basic research to understand who we are, what we're made of, and how we work. What's inside? How are ideas generated? How could you generate a sentence? Why don't any of the cats talk? But, another goal is, of course, clinical research. Once you know how a system works normally, then you're in a good position to fix it if something goes wrong. And our particular goal, especially in our lab, is to understand how the brain develops in the young child, and in particular, the role of input from the environment or experience in shaping brain development. This is very fundamental if we want to design educational curricula that optimizes human brain development. What could be more important than knowing when those systems in the brain that are important for learning and attention and language and math, when they develop and when they're most susceptible or needy of particular kinds of input. That's why we think that this is an important time now when we've learned enough about the human brain and brain development that we can start having a dialogue with educators like yourselves. We're there now. And we can do, these are all empirical questions, and all of these questions that you will have about education are tractable by the current empirical methods. Let me just remind you that in the adult brain, different regions are specialized to perform different and specific functions. So this is the brain from the side again, front and back. And in 99.9% of all the adults, actually all the mammals, this part of the brain at the very back processes vision. And this part up here in this fold processes hearing or sounds from the environment. In these parts of the brain mediate face perception in many animals. Of course, in humans, these regions are important for language. We know that certain areas in the frontal lobes are very important for emotional regulation and for social skills. And we know this first because in people who have had particular damage to this part of the brain are blind, basically. Or if they have damage to this part, they can't hear. If they have damage here, they have very great difficulty in regulating their emotions. We know this now, too, from imaging techniques that I'll talk about later. But what we're interested in, and what I think you're interested in, is where does all this complex, differentiated mosaic come from in development? And, you know, why are these regions at the back visual? Is it because there are genetic constraints that makes the neurons there capable only of processing vision? Is it a genetic constraint, nature? Or is it because in 99% of all the mammals, the eyes actually project there for some reason? And is it, on the other hand, that this is the visual cortex because visual inputs teach those neurons to become visual processors? That would be nurture. So this is just another way of stating the nature-nurture controversy. Why are things the way they are? Is it because it's genetically determined, or is it because the incoming inputs are instructive in setting up functional brain systems that they contact? It's not an either-or, it's an empirical question. How much of this is genetically determined? How much of it is pushable around? Very fundamental question for teachers and educators, and also parents and policymakers. We want to know where this differentiation comes from. Is it like that in the little baby? Well, it has been Brain Awareness Month. That baby has a brain in her head. Does it look like ours? Is vision at the back? Is it highly differentiated? Over what time course does it become differentiated? Well, we have learned over the past few years an astonishing fact, which I hope some of you have heard by now. I know the students in the room have, and that is that the human brain, just in terms of the hardware, the nuts and bolts, is not fully matured, it's not all there, until at least 20 years after birth. So it's not until the third decade. We know this by several different means. For example, Peter Huttenlocher has spent many years counting the synapses, those are connections, in the human brain, as a function of age. These are in brains that came to autopsy. And so, for example, in the visual cortex, what we see is that right after birth, there's a very rapid rise in the number of connections in the brain, in the visual cortex. And then there's a loss of about 50%. So the 2-year-old child has twice as many connections in her brain than her mother. And sometimes parents are horrified about this, and they think, what can I do to stop my child from losing their synapses? But it turns out this is a good thing. This is one of the major mechanisms whereby input from the environment or experience shapes a developing brain. And I think it's a good metaphor for this is that experience acts like a sculptor. So, you know, a sculptor starts with much more material, say clay or marble, than they end up with, but it's undifferentiated. And the sculptor takes away material to reveal a final form. And that is how experience shapes developing brains, by taking away the excessive or redundant connections and leaving a differentiated system. So experience determines which of these redundant synapses is going to be kept and which is going to be lost. Now, another very important fact that Huttenlocher discovered was that this overproduction and loss of synapses occurs at different times in different parts of the brain. So for auditory cortex, the curve looks like this. For the frontal lobes, which are the latest developing of all, it looks very prolonged and doesn't peak until several years after the visual cortex. So what this means is that the time periods in human development when input can maximally shape a developing brain system and the related functions is different. The brain systems develop at a different time. This mechanism whereby you change the brain is available at different times. There are different times when you can shape up different brain systems. We have to find out when those times are if we're going to be able to design educational programs that optimize human development. Now we have magnetic resonance imaging, and we have our own MRI machine on campus which produces beautiful images. We can look at the long time course of human brain development now with MRI. We don't have to count synapses in brains that came to autopsy. So this is just cortical gray matter, the cells. And you can see on MRI too that there's an excess amount of gray matter, and then there's pruning until at least 20, 25 years of age, when it finally starts looking adult-like. Now I'm going to start telling you about our own work in our lab, where we have many students and people working and our collaborators. And we're not looking at the development of the hardware or nuts and bolts of the brain, but we're studying the development of the functional specializations of different brain systems. And I'm going to tell you the bottom line right now. This is what we have found out after about 30 years of different kinds of research on brain plasticity. And that is that different brain systems show very different profiles of plasticity. There's a lot of variability. Some brain systems and related functions do seem pretty constrained. That is, they look the same even when experience has been very different. Other brain systems, it's very clear, are strongly modified by experience, and they're strongly dependent on experience, but only during particular time periods. And there are multiple different such periods, and these are what have been termed critical or sensitive periods. There isn't one sensitive period in human brain development. There are several. And furthermore, there are other brain systems which don't show any sensitive periods at all. They remain modifiable by experience throughout life. Now, these different profiles of plasticity don't occur just between different domains. Like, it's not as though the visual system is like this and language system is like this. Within vision, central vision is very constrained, but if you have a lazy eye, you have to correct it by 11 months, or you'll never have depth perception, so that has got a critical period. But if you have a scotoma in the back of your retina, the neurons that used to represent that part of the visual world will now reconfigure and represent something else. So within the visual system and within the language system, you see all these profiles. And I'll be illustrating that as we go along. And the way we found this out is we take two broad developmental approaches. One is what we call sort of retrospective or backward looking. We look at adults who've had a very different sensory or language experience. So people who were born deaf, people who were born blind, people who learn sign language instead of spoken language, people who learn English at the age of 12 instead of at birth. That's an important approach where you're comparing adult brains with adult brains. The other very powerful method is prospectively to study infants and children as they go through different ages and different stages of cognitive development to look at what changes are occurring. And this is a bit more challenging because it's challenging to get little children to sit still while you image their brain, but we can do it, and it's well worth it. Yeah, so one of the techniques we use, and especially with children, is we put on these hats, and we're basically, from these little pieces of silver that are sewn in these buttons, we're eavesdropping on the electrical signals that the brain is making when it's working. This is an electrophysiological approach. Of course, it's completely non-invasive. You just wear a hat, because the electrical signals your brain is making are conducted to the surface. We just pick them up and amplify them. And it's more particular than that. It's not just like the EEG. This is microvolts and this is time. And so this is the evoked response elicited specifically by a sound. And we can look at the events that occur in the brainstem, subcortically, and in the cortex. We can see how they're changed with attention and so forth. So we actually time lock our sampling of brain activity depending on what particular cognitive process we're interested in. It gives us great temporal information. We can learn exactly how quickly and when the brain is processing different information. When does attention make a difference? 1/10 of a second. The other technique we use is magnetic resonance imaging. We use this also with children, a little bit older children and adults, where you just, it's a head-only machine. You just put your head in there and we get these beautiful images. Beautiful, again, completely non-invasive. Of the structure of the brain, that's looking from the side. Of course, we can look at the function of the brain. So this is what part of the visual area is active by stimulating different parts of the visual field. We can look at the connections in the brain and even the chemistry of the brain. So That's why we think we're in a good position now to start making contributions to issues around education. I'm going to tell you first about work we've done to look at the effects of experience on perceptual processing. What happens in a person who's born deaf, completely deaf? What happens to auditory cortex? Does it just atrophy? What happens to blind people born blind? What happens to visual cortex? Does it just atrophy? Could it do anything else? How could that be? What does visual and auditory processing look like? And then we'll look at development of vision and audition. The deaf subjects, I'll just say a word about the ones that we've studied for a long time. They're all genetically deaf, because then the etiology is common, and it's because the eighth nerve doesn't differentiate properly in the inner ear, so the central nervous system is not affected. They're all congenitally deaf and profoundly deaf bilaterally. Now, we want to look at the effects of this auditory deprivation on brain development, but our subjects have also had really different language experience. They have all learned American Sign Language from birth from their parents, who are also deaf. So in order to separate out the effects of learning sign language on brain development and auditory deprivation on brain development, we study the siblings of these people who are born hearing, but who also learn American Sign Language as a first language. So any effects of auditory deprivation should be seen just in these subjects. Any effects of learning sign language will be seen in both groups. And here's the bottom line of what we have found in several studies of the visual processing after congenital deafness. The visual system in humans, it begins back here in the visual cortex. This is the back of the brain. But it has two major pathways. One projects up to the top of the brain, the parietal lobe. And this pathway is called the dorsal pathway because it projects up dorsal. And this pathway is very important for peripheral vision, for attention, and for motion processing. The other pathway projects forward in the temporal lobe, and this pathway is very important for central vision, for color processing, and for form processing. So it's a very differentiated two pathways within the visual system. And what we have found is that deaf and hearing people look identical on processing that occurs along this pathway, but deaf people are much superior in the kind of processing that this pathway does. So that suggests that this pathway may be most plastic, modifiable, enhanceable, but perhaps also vulnerable, and we'll look at some data for that. Okay, I'll just present some evidence for what I just said. So, brain responses to stimuli presented in the center of the visual field are the same in deaf and hearing people. But if you present stimuli out in the periphery, deaf people have much larger responses, larger than both normal hearing subjects and hearing native signers. So this is not an effect due to acquiring sign language, it's due to auditory deprivation. Deaf subjects are much faster and more accurate at detecting motion in the periphery than hearing people or hearing native signers. So there's a functional benefit in terms of behavior. If we look at the electrical potentials elicited by a color change, so here where the blue bars suddenly change to red, and compare it with the response elicited by when this grating moves to the right, the responses to color are the same in deaf and hearing people, but the responses to motion are much bigger in the deaf than the hearing people. This is all evidence for an enhanced dorsal visual pathway. We use MRI to look more particularly where in the brain these changes are taking place. What's changed in the visual system? Is it the case that the visual areas themselves are more active? Is it the case that multimodal areas of the brain, the parts of the brain that receive both, say, visual and auditory input, that those, in that case where audition is absent, the visual inputs could take over the whole structure? That's a reasonable hypothesis. And what about these auditory areas themselves, primary auditory cortex? What's it doing in a deaf person? Could it come to process visual information? And we use stimuli like these, which are either stationary field of dots or it's moving in and out. And when we looked in MT, which is the part of visual cortex that responds to motion, we saw very clearly that deaf subjects do have greater activity than hearing subjects, especially when they're attending into the periphery. but not when they're attending center. So the visual cortex shows enhanced activation. Multimodal cortex, like parietal cortex up here, is much more active for deaf people than hearing people or hearing native signers. So these effects are due to auditory deprivation, not due to the acquisition of sign language. So the changes in visual cortex, multimodal cortex. What about primary auditory cortex? Which is thought, as Mike said in his introduction, to be very genetically hardwired, these primary sensory areas. But what we observed, now this is a cross-section of the brain, like taking a slice like right about here. And this is the temporal lobe. Sitting right on top of the temporal lobe in red, shaded in red, is primary auditory cortex. And indicated in yellow are little bits of tissue there that are very interested in motion. They go up with visual motion, down when it's stationary, up, down, and so forth. And in every deaf person we've looked at, we see visual auditory cortex is very interested in visual motion. How could this happen? How could visual inputs, which are always going to the back of the brain, how could it get to an auditory area? That's a good question. We'll talk about that after I finish my story here. Now, so if this dorsal pathway is more enhanceable, more changeable, this raises the likely or reasonable hypothesis that it might also be a more vulnerable pathway, right? Because two sides of plasticity. A system that is changeable could be enhanced with good extra input. But what if the input is absent? That is going to be a more vulnerable system. And it's interesting that nobody had ever actually looked at both sides of plasticity. But we'd been reporting enhancements in the deaf, and then in a whole separate literature on developmental disabilities, like of dyslexia, autism, Williams syndrome, a couple of others, people have been reporting deficits in this dorsal visual pathway as well, but the two literatures were completely separate. And so Courtney Stevens in our lab did an experiment to see, using the same paradigm, could you show two sides of plasticity? And so she had deaf people sticking their head inside of this dome, which was extending out to here, and they had to press a button when they detected a light moving in from the far periphery. And just as we would have predicted, deaf subjects detected this movement in the periphery much sooner and significantly sooner than hearing people. And she did the same experiment with a group of dyslexics, adult dyslexics. And what they showed was exactly the opposite. They detected the moving dot of light much later than their matched control group. So this shows the two sides of plasticity, systems that we have now identified in people who've had quite extremely different experience, deaf people. Those are systems to look out for and target, perhaps, when we think about identifying systems that might be most vulnerable in normal development. I'll just say a little bit about work we've done on blind people to look at parallel issues within the auditory system. For example, we did an experiment very parallel to some studies that we did in the deaf with an array of speakers in the front and in the periphery of auditory space. And we had blind or sighted blindfolded subjects sitting here, and they had to detect targets either coming from speaker one and ignore the targets when they came from these speakers, or they were attending to the periphery and detecting targets here but ignoring them when they came here. And we wanted to look at the accuracy of sound localization in the center and the periphery. And what we found was that in the center of auditory space, blind and sighted people are the same. Just like deaf and hearing subjects were the same for central vision. Looks like high precision systems like central vision, central audition are pretty tightly constrained. But the blind people were much better and more accurate at localizing sounds to this speaker in the periphery than were the sighted subjects. Also, if we look at rapid auditory processing, like the ability of neurons to recover a full response, even when sounds are coming very rapidly, as they do in normal speech, by the way. Whereas with rapid speech, we tend to have a pretty small response. Blind people have a much enhanced response. So they have a shorter refractory period or more faster recovery of the auditory system than we do. Well, let's look at the other side of the story. we know that rapid auditory processing, as we'll hear from Mike Merznick tomorrow, has been implicated. Deficits in rapid auditory processing have been implicated in language impairment and dyslexia. And in this same paradigm, where we present tones at different rates, language impaired children do show considerably reduced responsiveness compared to normal control groups. So this is two sides of plasticity. Systems that are enhanced in the blind may be at greater risk in children who are vulnerable. And just one more thing about the blind is, you might ask, well, what's this huge visual cortex doing in the blind? Well, we presented auditory sentences to sighted and blind subjects, and you know, this is a side view of the brain again. When we listen to auditory sentences, we activate Broca's and Wernicke's classical language areas in the left hemisphere, but we don't activate visual cortex. But blind subjects, they activate Broca's and Wernicke's area, but they also activate visual cortex. That's right in V1, because Brigitte Roeder, who did this experiment, identified the calcar and sulcus. So another piece of evidence suggesting that these anatomical areas, visual system, auditory system, these aren't genetically defined or anatomically defined, they're functionally defined. But you're still wondering, well, how did you get auditory input into the visual cortex? How did you get visual input into the auditory cortex? And there's several different possible mechanisms. Each would be available at a different time in development. This is important to know when you're considering doing a cochlear implant, for example. By which mechanism could a cochlear implant sort of reinstate auditory processing in the auditory cortex? Well, one is that you could have very altered subcortical to cortical connectivity. So in normal mammals, the ear projects to auditory structures in the thalamus and then to the auditory cortex. But we now know that there's a time early in development when mammals have all those extra connections, when there's a small, somewhat weak connection from the ear, actually, to the visual structures in the thalamus. And it could be that this normally transient, redundant cross-modal connectivity is strengthened when there's no competition from visual input for that area. And there's evidence for this in animal models. Very good evidence. But obviously the time when this mechanism would be available for rewiring the ear into the visual cortex would be very early in development when we have those extra cross-modal synapses. So that's not a mechanism that would be available throughout life. Another kind of mechanism would be changes in cortical-cortical connectivity. For example, there is a connection from primary auditory cortex to visual cortex that's just been discovered in the past few years. And it turns out it's not just anywhere in the visual cortex. This auditory projection goes to peripheral representations in visual cortex. And it could be the strengthening of these cortical-cortical connections is what occurs in our deaf subjects and accounts for the special plasticity of the periphery. Now, another mechanism that would definitely be available all throughout life is cortical-cortical feedback, like that occurs when parietal cortex talks to visual cortex and directs attention toward an object and actually changes processing in visual cortex. And I'll show you some data that shows how that works and how quickly that works. So attentional mechanisms, which are importantly organized within parietal cortex, are available throughout life to enhance visual processing. What's the likelihood that we have cross-modal connections early in brain development? We know that they're there for other animal species. We have some evidence that they're there for us too, because we presented auditory stimuli. Now, auditory stimuli produce a good, crisp brain response over temporal cortex here in adults, but not over the visual cortex. But if you look at newborns, auditory stimuli activate both temporal and visual cortex. But gradually, the auditory response over visual cortex gradually is reduced until by 3 years of age it's gone. So this suggests that maybe the first 3 years of life is a time when this kind of rewiring can occur in a person who's congenitally blind or deaf. This would be important information when you're contemplating when to do cochlear implants and so forth. Another mechanism which might confer more plasticity on a system is if it had a longer developmental time course. And so one way we looked at this is by looking at the motion and color processing systems using those same color and motion stimuli I described before, because we noted that the color responses didn't change much, so maybe they had a shorter developmental time course, but the motion responses did change. And what we observed was that, in adults, the motion stimuli elicit responses that are faster than the color stimuli. And in neural processing times, this looks like a small difference, but it's a big one. It's about 20 milliseconds or 30. But even at 8 years of age, we don't see that pattern. At 8 years of age, whereas the color response of these kids is on time, the motion response is really slow. So this suggests that a longer developmental time course may confer greater plasticity on a system as well. That could be another mechanism, in addition to the redundant connectivity. What about attention? Well, I know so many people think that attention is like just a psychological construct, or like Mike said, just a new age idea about something. But it's very real. It has a chemistry, an anatomy, and a physiology that we know very well. We have attention in a jar, thanks to Mike Posner largely, who's really a pioneer in this field. Let me show you the effects of attention. So if I have you staring here, and I flash little lights either in the right or left visual field. But the experiment is I'm going to ask you now either attend to the left visual field away from the stimulus, or attend to the right visual field where the stimulus is. Here's the brain response if I tell you, attend over here. Pretty small. That's a little brain event. It's about 1/10 of a second after this comes on. Now, if I ask you, okay, just move your attention, not your eyes, by the way. Your eyes are always focused here. You're just moving your attention over here. And when you do that, the sensory response is amplified at least 5 or 6 times. It's huge. Big change in sensory processing with attention. That's how powerful attention is. And look how fast it's operating. 1 to 2/10 of a second after that stimulus comes on, your brain sensory processes has changed. Now, we looked at this in deaf subjects. And deaf subjects, too, when they're not attending, when they're attending away, you get a small neural response. But when they're attending, they get a response that's even bigger, an attention effect that's almost 2 to 3 times as large as that in hearing people. That's shown here. So hearing people, our sensory response can be increased up to 100% with attention. But in deaf people, it can go up to almost 200%. So, of course, we're interested in studying the development of attention in young children and looking at the effects of training attention in young children. So, it's hard to get children to stare straight ahead and move their attention around. So we designed another experiment to try to get at these same mechanisms. We present children with 2 stories at once, the blue kangaroo and Harry the dog, say. And they're played out of 2 speakers at the same time. It's astonishing to hear for anybody. Now, but we say, but you only must pay attention to one story. And then there are pictures on the screen that go along with the story that they're supposed to be attending to. And then, so blue kangaroo, listen to the blue kangaroo. And the stories move around from speaker to speaker, but they have to move their attention with the story. Now, superimposed on the story itself, which is shown here, we have little buzzes and baws. And these are like probes that we're going to record the sensory responses to and see if they're changed by whether or not the child is attending. Of course, the first thing to do was do this in adults to make sure we got the same attention effect, and we did. So this negativity is much bigger when those adults are listening to the story than when they're listening to the other one. And children show a very marked effect of attention by 1/10 of a second. So when children are attending the stories, this positivity is much bigger than when they're attending to the other story. At 8, 7, and 6 years old. Now, we've just analyzed data from a group of 3 to 5-year-olds, and you see exactly the same effect. It's very robust. It's on time at 1/10 of a second. Of course, it looks very different from the adult because these are just baby brains and they're still developing. And the adult effect has got this shape, and the baby one is like this. But still, attention is modulating sensory processing at a tenth of a second. So attention is very plastic. We saw that in the deaf. There are other studies of animals that have shown that. It's actually very important for neuroplasticity. I didn't have time to talk about that. And it develops over a long time. That's just a summary of what I said. There's increased performance in the periphery, auditory periphery for the blind. This pathway might be more vulnerable. Fast auditory processing appears to be more vulnerable. We talked about mechanisms for plasticity, including differences in redundant connectivity, differences in developmental time course. There are differences in the chemistry of the dorsal and ventral pathways as well, very clear ones. And also, it's important to talk about functional demands and attention. So obviously, we as hearing people rely very importantly on our hearing to identify potentially important events in the periphery of our world. Deaf people have to rely on peripheral vision to monitor the peripheral world. So they're always probably directing their attention to the periphery more than we are. So all these different factors are going to be important in determining a particular system's plasticity profile. I'm going to talk a little bit about language processing now. When you look at the visual system, some parts are more plastic than others. You look at the auditory system, some parts are more plastic than others. What about language? It has to be like that too. There are many different subsystems in language. Which ones are the most dependent on experience? Which ones can change throughout life? And we've addressed these issues by looking at bilinguals, people who learn signed instead of spoken language, and a lot of individual difference data that I'm going to get to. We look at semantic processing in language, the process whereby words stand for particular objects and events, and grammatical processing. One of the ways we do that is to look at sentences which have a semantic surprise in them. That's a normal sentence, grammatically and semantically normal. This one has a semantic surprise. The man bought Tom's wink. You don't buy a wink. And this one's semantically okay, but the words are out of order. The man bought Tom's of painting the ocean. So these are some of the ways that we eavesdrop on the brain when it's presented with a semantic surprise or a grammatical surprise. And sure enough, now this has been replicated in many, many labs across the world, that we all use posterior systems, this is the nose, of both the left and the right hemispheres when we do semantic processing, and we use systems in the anterior region of the left hemisphere to do grammatical processing. This is in adults. Well, now you can say, well, what about people who learn English late? Are semantic and grammatical processing affected equivalently? And Chris Weber-Fox did this study where we looked at people who came from China to America, either pretty early, 1 to 3 years of age, 4 to 6 years of age, or pretty late, 11 to 13 years of age. And in terms of semantic processing, everything looked just the same. The semantic surprise response was identical, regardless of age of acquisition of English. So this suggests that the semantic system in language is very robust, capable perhaps of change throughout life. And in marked contrast, when we look at grammatical processing, whereas native speakers and early learners of English show this system in the left hemisphere, not the right, when they're confronting a grammatical error, even a delay of as few as 4 to 6 years results in a more bilateral activation pattern. A delay of longer, like 13 years, results in a very aberrant, for oral languages, this is a very aberrant pattern. And behavioral test scores of these same subjects show very marked decline with increasing age of exposure to English in grammar. Many studies have shown the vulnerability of phonology. You need to be exposed to the sounds of a language early if you're going to speak with a non-native accent. You all know that, and it's been documented by many studies. I mean, even a delay of 5 years, and people don't sound like native speakers. This is native speakers. This is their rating of sounding native-like. It's just a linear decline. So phonology and grammar, you need to get them early. That's why we need to get second language education in the elementary schools instead of saving it for high school. You know, little children are avid language learners. They can learn 5 languages like this. And the more you put it off, the harder it gets. So what's the reason of saving second language education for high school? What about deaf students? What about American Sign Language? This is interesting pragmatically, but it's also interesting, I think it allows us to address a fundamental question, and that is, why is it that the left hemisphere is ubiquitously, all around the world, the more important hemisphere for language? Why is it? And some people have proposed that it's because the left hemisphere is a very good rapid auditory processor. Well, if that's the reason, then we shouldn't necessarily see recruitment of these classical language areas for American Sign Language, which is not auditory. Indeed, sign language is visual, uses space, visual space for grammar and the lexicon. We know the right hemisphere is very important for visual spatial processing. So we've done quite a bit of work looking at the organization of the brain, comparing it for written English and American Sign Language. Now, let's look at the results for English. We had 3 groups of subjects. Normal hearing people. When we read, we very strongly activate these classical language areas in the left hemisphere, Broca's area and Wernicke's area. But it's very left-sided, not much activity on the right. Now, deaf subjects, when they read English, and they learn English late and imperfectly, they don't recruit these classical language areas. You see activity in the right posterior regions. Is that because they learned a visual language first? No, because their siblings, hearing native signers, who also learned ASL first, don't show that. They show a classical hearing pattern of recruiting Broca's area, and Wernicke's area was below threshold in this image. Okay, so why, the real key question, why don't deaf people recruit the left hemisphere language areas? And if we knew what factors were important in recruiting these areas, this would be importantly inform us about language education. Is it because they learn English late and imperfectly, or is it because they learn a visual spatial language and they don't learn English as an oral language? Well, to ask this question, you have to look at American Sign Language. Normal hearing subjects don't show any, who don't know ASL, don't show any difference in activation between signed sentences and non-signed gestures. You can't tell the difference because you don't know sign language. Deaf people, when they look at their native language, they recruit Broca's and Wernicke's area very strongly, very robust activation. Now, they also recruit homologous areas on the right side of the brain. Is that because they're deaf and they have a hyperactive visual right hemisphere? No, because the hearing native signers also show that pattern. They look identical. They recruit left hemisphere for sign language and right hemisphere as well. So what I think is interesting about this is it suggests that there's a strong biological bias for these regions of the left hemisphere to mediate human language, any formal human language, regardless of the modality that it's instantiated in. So that suggests there are biological universals of human language. It doesn't matter if you're hearing or deaf, reading English or sign language, you always recruit those areas. But clearly, experience, language experience, plays a very important role in the final configuration of the language systems. Because if you learn a sign language, you strongly recruit the right hemisphere. If you learn an oral language, you don't. Now, is there a critical time or a limited time when people could recruit the right hemisphere into the language system? So Aaron Newman looked at people who had learned sign language either from birth, and where you do see a lot of right hemisphere activation, or people who learned late at the age of 18 to become sign interpreters, and they don't show this activation in the right hemisphere. So it's better to get ASL early if you want to recruit an optimal language system, and of course behavioral tests have shown that late learners of sign language don't know the grammar of sign language as well as early learners. So children have to get language early. It doesn't matter what modality. Actually, deaf people who learn sign language actually do better on tests of English as well. They can use sign language to learn English. Another study that Cheryl Capek did for her thesis, this is getting at the question, a lot of people say, well, there's no maturational constraint in the brain that makes it less able to learn language late. It's just that your first language interferes with your second language. That's why it's harder to learn language late, right? Well, that's very difficult to disentangle. But you can if you look at deaf people, because many deaf people don't learn a first language until late. Deaf people who are born to hearing parents, who just keep talking to them, and then maybe they only finally learn language when they go to school or middle school. So what we did was look at people who learned their first language at different ages to see if the deficits that occur with late learning are totally the result of language interference or whether there are maturational constraints. And when you look at semantic processing, which was a very robust system capable of change throughout life in bilinguals, we also see that in the deaf people, they can pop off this brainwave associated with semantic surprise perfectly normally, even if they don't learn their first language till 15 to 21 years of age. So that semantic system can be set up anytime. But if you start looking at the grammatical system, whereas early learners, those who learned basically before the age of 10, they do show this little effect of syntactic surprise, but late learners don't. So there are biological constraints that make the brain better language learner early than late that we need to be aware of. Just a summary now, and that is that just what I said. Some systems are constrained, like high acuity systems, the left hemisphere. Some are modifiable, but just within particular time periods. Some display lifelong plasticity, and Mike Murdnick did a lot of this work showing that the representation of sensory maps changes throughout life, lexical acquisition. And we're always trying to think about what the invariance is with systems that display these different profiles, but we're not really there yet. Now I want to talk about individual differences. We've been able now to identify which systems are more or less likely to be plastic and vulnerable. And we're looking at them now in just normally developing people by studying little kids like this. And one way we can look at the effects, well, individual differences, is what we've observed is that at 20 months of age, so in a 1.5-year-old child, they're already starting to show a left hemisphere asymmetry for when they listen to words whose meaning they know, left, not right. A typical 1-year-old doesn't show that effect here. A typical 1-year-old. But if we look at kids who are the same age but have big vocabularies, they already show the beginning of that effect. So so the left hemisphere specialization for language gradually emerges. It's not there at birth. It gradually emerges. It doesn't just show up on time at a certain age. It's importantly predicted by language knowledge. We've seen this also in sentence processing. This is Amy's thesis work, Amy Harris. So, well, this is interesting. Even 3-year-old children already have different brain systems mediating semantic and grammatical processing. So for semantic surprises, they get this posterior left and right hemisphere response, but for grammatical processing, they show this left, not right hemisphere response. 3 years of age, that's interesting. Every 3-year-old? No, she gave these kids tests of knowledge of English grammar, the preschool self, and the kids who scored high in the top quartile, they show this left hemisphere bigger than right effect for a syntactic surprise, but the low comprehenders didn't. Now, Eric Pakalak, who's here, said, gee, I wonder if you'd see such differences even in adults between native speakers of English, monolingual native speakers of English. Would you see much variability in their language knowledge? And you do. Of course, if you give them tests of grammar, some score pretty low in the 25th percentile, others score high. These are just 3 different tests of grammar. I wonder if their brain organization is different. And so Eric looked at the effects of a syntactic surprise in the high proficiency people. And there you see just what you'd expect, this negativity, left anterior negativity over the left hemisphere, not over the right hemisphere, specialized left hemisphere system. But if you look at low proficiency adults, native speakers of English, well, you get this effect, but it's everywhere. It's over the right hemisphere and the left. So it's just a much less specialized language system. and people with low proficiency. Okay, now, where are these individual differences coming from? Of course. Well, there's two ideas, right? Well, we know one thing. We know from Hart and Risley, who did a study just counting the number of utterances that parents spoke to their children, that there's a big variability in the amount of language input that kids get. So some parents are very talkative. What they did is just count the number of utterances per visit, and they visited kids for a year, like once a week. And this is the age of the child, and it's pretty constant. Some parents are very talkative to their children. Some are moderately talkative, and some are not talkative. And if you look at the vocabularies of these very children, of these parents, starting at 18 months of age, there's a huge difference, such that those kids who have talkative parents have a big vocabulary, moderately talkative a moderate one, and not talkative a low one. Now, they've followed these kids out now to 10 years of age, and these differences are maintained. It's a long time, big effect. And of course, we know that these kids with big vocabularies have more mature brain organization than these kids, because we've studied that, the effect of vocabulary size on brain organization. So you could say from these data, well, that just shows the importance of the environment on brain development, right? Not necessarily. I mean, children and adults share both environment and genes, right? Maybe talkative parents have talkative children. We certainly know that genes are important in brain function. They've been identified, genes that are important in predicting individual differences in attention, memory, spatial skills, novelty seeking. Not quite there for language, I don't think. We know that gene expression is very different depending on the environment that people are in. I don't have time to go into that. We know that environments are hugely variable. Like if you're born in the United States and your parents have a good education and a good occupation, high occupation, you yourself are likely to have high language, literacy, numeracy, health, and longevity. But if your parents had a low education and a low occupation, you're very likely to be low on these very values. Now, so environments are highly variable. The effect of environment is different in different countries. It doesn't have to be like that. Like if you're in Sweden or Canada, you know, things are not quite as severely different. But anyway, environments are different. Low SES environments differ in so many variables. Right. These are all of them. We've looked at kids from low and high SES, and other people have too. And it's very clear that low SES kids have lower cognitive skills and they have less mature brain organization. The problem is, you can't really talk about that, I don't think, because you don't know the reason. It could be low SES people are low SES for some biological reason. That's what some people say. And of course, it could be they're at a cognitive disadvantage because they have very disadvantaged environments. You have to sort it out if you want to go to the policymakers and say, I need more money because I know that experience is the factor, right? So you have to sort out genes and environment. A correlation does not mean a causation. Of course you knew that, but it's a trap we keep falling into too. We have to watch out for this all the time. So what are we doing about this? Well, first of all, I'll tell you a couple of other people's studies that address this question. So Jane Ellen Huttenlocher did a study to look at the effect of teacher language on child language development. So teachers and children don't share genes, right? And what they found was that teachers who produced more complex sentences, so this was number of noun phrases per sentence, well, their children's language grew more over the course of a school year than children who were in classes of teachers who use less complex language. That's a very important study, I think. Another important group of studies is being done by these pediatricians who belong to a group called Reach Out and Read. And these pediatricians typically work in very low SES neighborhoods. The minute they're in the newborn nursery with the newborn baby, they start talking to the parents about the importance of reading to their child. And they give them a book. And at the 6-month follow-up, they talk to them about the importance of reading, and they give them another book. And the kid has 10 books by the time he's going to kindergarten. and he's red. And somebody did an experiment to see if the receptive vocabularies of kids in the low SES neighborhoods whose pediatricians belonged to this group and kids who went to pediatricians that didn't belong were different, and they are significantly improved. The kids whose parents read to them because the pediatrician told them to were significantly improved. So this is taking biology out of the equation too. Environment does make a difference. The other thing that we're doing is now interventions, as Mike mentioned. So last summer, we had a group of 6.5-year-old low language kids, and we saw them for 6 weeks for 100 minutes a day, and they used fast forward, the technique that Mike Mersnick developed that he'll talk about tomorrow. It wasn't regular fast forward because each of them had a one-on-one coach. So this is pretty intensive. But these kids were in trouble. They were in the 5th percentile for language. And after 6 weeks of 100 minutes a day, their language skills went up to the 20th percentile. 6 weeks, intensive. So these kids are trained not only to play language games, but to really focus their attention. They had to focus 100 minutes a day. And we thought that this might have a significant impact on the development of their attentional skills. And what we observed was that whereas normal kids of this age, 4 to 7, have a very beautiful increase in sensory processing with attention compared to unattention, these language-impaired kids did not have any attention effect before, but after they did have a very significant one. So this shows that in a relatively short time, 6 weeks, you can change language and change attention with 100 minutes a day. What we're doing now, ultimately what we want to do is compare different kinds of interventions in both low and high SES children. Our hypothesis is interventions will be more effective in low SES children. High SES children have already got very optimized environments. But now we're starting with low SES kids. And exactly what we're doing now is we're looking at children who are enrolled in Head Start preschool. So these are 3 to 5-year-old kids. These are programs for disadvantaged kids, low SES. And what we did is we randomly assigned children to an intervention which was 40 minutes of music a day, 40 minutes of language, 40 minutes of attention training, or their parents received training on how to be responsive parents, or a control group. We have a couple of control groups. And what this involves is we have actually a teacher now. So this is not computer games. Our teacher, Scott Klein, and he has an aide, and he takes 5 children, so it's a small group. 5 children, 40 minutes a day, 8 weeks. And we test their brains and behavior before and after. We test them, huge behavioral battery, of course, lots of aspects of language, math, attention, and so forth. And well, we've just done our first post-testing, and we begin our second group of post-testing tomorrow. So this is early in the days, but we're very excited because the kids who got the music training for 8 weeks, they improved, every one of them improved in math and attention. Language didn't change much. The language training improved in many different aspects of language and math. That didn't change. And then I think the most impressive thing was the group that got attention training. They were Scott just trained them by playing lots of little games just to focus. And they improved on everything. They improved on language, math, attention. The group who got parental training with Jessica, their language improved. That's just what she was hoping. Their math actually improved as well. But, you know, she talked to the parents a lot about sitting down and talking to your child and listening to your child. So, and the control groups are basically just not budging. This is small. These are only 30 kids, but we're starting the second group tomorrow, or today, I think is when we did it. This is with Whitaker Head Start. The other one's with Holly. And then we're going to go to the Head Start on campus, and we'll go back to Whitaker. And we'll be doing this for a few years to get enough data. But we feel like if we can go to Kulongowski or whatever, with a picture of a brain on good education and a brain without it, well, it might amplify, it might amplify the effects of behavioral test scores, which are obviously important, but You know, pictures of brains are pretty catchy these days anyways for policymakers, it seems to me. So, just to summarize now and end, specialized brain systems are important for every aspect of sensory, cognitive, and social emotional functioning. I didn't talk much about that at all, but those systems are biologically based too, and they are very dependent on good early experience. We know that from work that other people have done. The human brain develops over a long time. Experience drives and shapes the development of these systems. Optimal brain development requires adequate stimulation and nurturing is harmed by drugs and neglect. I didn't really talk about that. So what we have our work cut out for us, those of us who are continuing research. We have to determine the time periods of plasticity of other systems, the mechanisms that determine the plasticity, the interventions that can enhance them. And this is our toughest job, I think. To convince the public and policymakers to be guided by evidence. Any kind of evidence. Brain evidence is good, but any kind of evidence. Education is not an evidence-based domain of inquiry at the current time. People have said, you know, you have to be evidence-based somewhere in some document. It's written, but it's not the case. And I think you all know that, all of you in education. And then, after that, all we have to do is design and implement evidence-based educational programs for everybody. And this is very wordy. It just basically says, you know, this makes good economic sense, too. I mean, you know, the federal government is paying us millions, us in developmental cognitive neuroscience, millions of dollars every year to do this research. And they never asked us what we found out. You know, come on. You got to get your return on your investment. And it would cost a lot to do, I think, probably, to have an evidence-based educational system, but the long-term benefits on growing the economy of Oregon, for example, would be huge. I've got to get an economist with a spreadsheet on my team soon, get the numbers. So one thing that we're doing, I wanted to tell you about, is that my students and I are making a DVD which documents basic facts about human brain development and evidence showing the effects of experience on the development of vision, hearing, motor skills, language, reading, attention, music, math, and social and emotional systems. This we intend to provide freely available, downloadable from our website. It's not ready yet, but it will be ready. We really think it's going to be ready by the summer, by July or something. So go to our website and check it out if you're interested, and then you can just give it to people. Because I think one of the main difficulties is that, you know, a lot of people just don't know anything about brain development. It's like just getting the word from the ivory towers into the streets. Well, I know that's true, because my pediatrician didn't know that you have to correct a lazy eye by 11 months. He said, well, I did it at 4, and the surgery was perfect. I said, well, the neurons need to know by 11 months. They need convergent input. And he just didn't know it, you know. We do have a difficulty, I think. We need to develop, I should say, an interface, a strong interface between basic research and the real world, between, you know, basic medical science and physicians, between basic brain research and educators and parents. And parents don't know that you're supposed to talk to your children. I've had many parents say to me, well, I didn't know you're supposed to talk to him. Like, he doesn't understand a word of English. That's how he's going to find out. No, I'm serious. And I'm not being horrible. I mean, you just need to know. Well, I didn't know that you're not supposed to shake him. So this is our lab. Thank you very much.
If I could ask our friends in Bend, can they hear me? Can you hear me in Bend? Usually I'm loud enough to get over the mountains. Could you raise your hand? Is there a question there for Helen before I turn it over to the Eugene audience? Is there someone who wants to ask a question there in Bend? I haven't heard a question yet.
Time to go home, I think.
Let me try Eugene.
Well, you don't think they're hearing it, Ray?
Did I see a hand up?
I don't think they might not hear it.
We can hear you. I just don't think anybody here has a question.
I can hear you, so if you have a question, please ask when you have.
They said they didn't have any questions.
Okay, let me try one from here in Eugene. There's some questions here. Well, let me start off with one.
Well, there's a guy at the
Oh,
back.
there's one in the back.
You showed a slide which indicated that talking to children, those children of talking parents tended to be more talkative, have higher vocabulary. Are you also implying that the simple act of talking, beginning early with builds some kind of foundation not only for language, but for all these, what we call intellectual functions in the brain? Is that really some of what you're getting for?
Let me just repeat the question, just so that they be sure and hear us also other places. So are you implying that talking to your children also sets up the foundation for learning other cognitive skills beside language?
Well, that's a very good hypothesis, and it's one that we're testing now. I mean, it needs to be tested. So, in other words, will our language intervention with the little kids, will that improve nonverbal IQ? Will it improve critical thinking skills? Will it improve attention and so forth? And that's an empirical question. It's a very important question. You know, if you had limited time, I mean, some people have said to me when I told them these results, they said, well, how long is that going to last, your little 8-week thing? I said, no, it doesn't matter. Why don't you just provide for the capability so that each child can be experienced in a small group for 40 minutes a day. That's it. Just do it. Now, if you were going to do that, so each child could be in a small group for 40 minutes a day out of the school day. I mean, how difficult would it be? But then you'd have to decide, what are you going to do in that small group? What's going to be the most important thing you could do? Is it going to be language? Is it going to be music? Right now, I've got my money on attention. But we'll see.
There's another question in the back. Yes?
Yeah, I have a question. When you talk about interacting with your child, do you mean talking to your child? Is there a difference between talking and reading? Or how would the book on tape be? It doesn't have to be interactive, or could it be just listening to language? Is there a difference?
There is a difference.
Let me just repeat this. The question is, does it have to be an interaction? Does it have to be language? Or would any interaction do? Or could you just turn on a tape or a television?
Oh, well, you know, those are the kinds of things that you wish somebody would do a study on, and somebody did do a study. And children learn much better from a live human person than from a tape. And the other thing is, this needs to be quantified too, but part of the reading to your child thing, you know, because a 9-month-old kid is not reading, you know, but what everybody believes to be the case in this business is that the act of sitting with your child on your lap and you talking about the pictures, it's not just reading, like it's the whole interaction and the give and take. And, you know, your child is pointing out this and you respond, and you say this and your child responds. So it's this whole kind of social discourse, and language comes along as well. But, yeah. So,
Another question in the center there? Yeah.
I was wondering if we get some examples of the attention exercises that we use.
Some examples of the attention exercises that you used.
Scott did so many things. We all sat around and talked to a lot of people who had been thinking about this and got input from a lot of people. So we do a lot of different things like, look at this strawberry. What color is it? Red! Do you see any other colors in there? Black! You know. Oh, any other colors? Oh, oh, yellow. And, you can only do that for so long. And then, so you have to be pretty ingenious, okay? And then another one is just, okay, just try moving your muscles like this. Pay attention. How does it feel? What does it feel like? Put that one back. Attention to your muscles. Attention to a sound and a not sound. They'll make a big noise and then be really quiet. You do some yoga exercises. But the good thing is that this summer, Scott, our teacher, is going to document all of these, everything. And we're going to put it on our website too, so that it'll be available. Yeah.
I think there was a question. Did you have a question?
Oh, that was my question about the attention.
Oh, okay. Is there? Yes.
Yeah, and in addition to that, is it possible to do those with older children? Would there be a result, do you think?
Yeah, but that's such a good question. Of course.
Just, I can just repeat the
Yeah.
question. Is it possible to do this kind of training with older children?
Of course. And, huh? No, but that's an important question. I mean, that's the critical period question, right? So we started young, low SES. We hypothesized that's where we can make the biggest difference. But it's an empirical question. So we are also doing some studies with 6 to 8-year-old kids. And then ultimately you'd want to do them with older kids. And then you have to... Yeah. Well, I know. But, how many... Well, I have... there's a student, Sonia, but she's not here. She's going to start doing an intervention with teenagers on attention that we're going to talk to her about it. She's a clinical student in our department. But so people are going to look at that. I mean, it takes a lot of people to do this. These intervention studies, oh my God, it takes so hard.
Other
They're
questions?
so hard.
Right here? Yes.
So, is there ever too much? I mean, I have a yuppie sister, and each of her kids had 16 mobiles above their crib. And she will never let them walk around the house without talking in their ears, yap, 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 all the time. She never does it. You can't have a conversation with her because she's always talking to her kids.
Yeah.
Question is-
plateau, or does it get too much?
The question is, can you have overstimulation, too much input?
Well, of course, I haven't done a study on that. And, I always want empirical evidence, but of course, that's distracting, right? I mean, that's the opposite of attention in a way, is to have multiple distractors going on at once. I mean, if the child was forced to pay attention to just, her sister's voice and ignore her mother's voice, maybe that would train attention, but somehow I'm getting the idea that she wasn't helped in doing that, right?
Yes. Kevin?
When social scientists look at abused and neglected children, they study the construct of resiliency. I'm curious to hear your thoughts about the connection between brain plasticity and resiliency.
So the question deals with the questions between, the relations between brain mechanisms and resiliency as it's studied by people who study abuse and neglect.
Well, people, we're learning so much about, we've actually learned a lot about the effects of stress or lack of nurturing or neglect on brain development. They're huge effects, of course, just huge. And they're good animal models of this. I mean, it affects gene expression in your brain, for example. And, most of us in this field really recognize that probably social emotional development is, the most important aspect of early child development of all. We didn't talk about it so much tonight, but that's because it's not my primary area of research, although the parental training thing is really getting at that. I mean, that's what that's about. Talk to your child, pay attention. But anyway, I'll just tell you another factor that's been identified. Resilience, what would make for individual differences in response to abuse. And there's very important study published in the last 2 years showing genetic differences, actually. And this is in the serotonin transporter, for example, and the MAOA gene. And so, for example, there are 3 different forms of a gene that's important in transporting serotonin. And you either, depending on what form you get, you either have a high, a moderate, or a low degree of serotonin. And if you look at people who've had terrible, or 1, 2, 3, or 4, or 5 awful life events happen, and whether or not they're depressed, well, it's really quite predictive by which gene allele they had of the gene. So that's what I meant by the importance of gene-environment interactions. Now, if people who had different forms of the gene that had one or two low stressful events, didn't have very much, they were all fine. So if we could just optimize the environment to where everybody's nurtured, nobody's neglected and abused, then these allelic differences wouldn't be a knob that turns anything. But it turns out it is an important factor when there has been abuse. This is also true for child abuse, for predicting whether or not a person is going to abuse their child if they were abused.
Yes.
Two questions that are sort of unrelated. But those attention exercises, it sounds like you're systematically exercising the different sensory systems, starting with the colors on the strawberry and hearing loudness and softness and 10 different parts of your body and doing yoga. So how much of that is attention versus exercising the sensory systems? And can you take the attention training separated from the sensory training? It makes sense if you systematically train the different sensory systems, you would improve in many different cognitive areas. And is that really attention or sensory exercise?
The question is whether this is attention or sensory training that's involved in your when you're doing training of attention.
Oh, no, they do imagery as well. It's not just all sensory. So they say, okay, I want you to imagine, your house, and I want you to count how many windows you have in there. So,
that's also systematically training different functions that are, how's that attention, and can you do attention separate from those things?
well, for example, well, you'll have to see all the exercises that he does. But what we're trying to train up actually sustained endogenous attention. I mean, the word attention is used in many different ways. But what they have to do is keep their attention focused and still on one point for a long time.
There was another question.
Follow-up to that, and I think the common pattern there is language. Look what you're doing there. It's really hard to separate language from attention. And to me, it sounded like very good language stimulation. You're creating interest in the child, and you're talking about a lot of different things and keeping their attention by changing topics. So it's really hard to separate out language from attention.
Question is, can you separate language processing from attention in your exercises?
Well, we have to use language in each of the interventions, of course, and to some extent we use attention in each of the interventions, like with music, when the kids are making music and making stories and so forth. And we just tried to keep things as pure as we could, but we did have to talk to the kids. And the music, whereas the language groups really did improve in their language skills, phonological awareness, vocabulary, and so forth. The music kids and the attention kids, that's not where they went up.
There's a question here.
Yes, you indicated on the graph the up arrows and the down arrows. Fortunately, no down arrows. So which ones of these arrows could you speak to now and amplify in terms of really significant shifts in the up?
The question is, which of the changes indicated in the graph for training were significant statistically?
Oh, I wouldn't have put an arrow there if they weren't statistically significant. And the reason I'm even talking about this, because it's pretty preliminary, is because it was a huge effect, like every kid in the group. It's either every kid in the group or every kid but one that went in one direction. Otherwise, I wouldn't be talking about it. But it'll be a long time before we can publish because we have to get more kids in every group.
Yes, one more question here.
Do you think you could get the same effect on kids that were in a more typically developing children? Do you think you get the same effect with those exercises?
Question is, would the same effects of training occur with more typically developing children?
I don't. Well, that's an important question that we want to look at. And as I said, we hypothesize that the effects of interventions will be greater in low SES children because high SES children already have pretty optimal environments. But that's an empirical question. We need to do those studies. But we started with a group that we thought would give us the biggest chance of finding a difference, young, low SES. and then we have to do everything else. You can see this is going to take a while.
Are there additional questions from the Eugene group here? Yes, over here, and then this one. First one here.
Do you think that training that you did would have had the same effect if you were using larger groups, like maybe 30 kids, as a typical classroom?
Helen, could you repeat the
Yeah,
question?
the question is, would it occur if you had larger groups? That's an important question too, and it's an empirical question. And, you know, we have to do the experiment. We wanted to optimize our chances of making a difference, so we had a small group.
Another question here.
So you were talking about the research in the social and emotional area. I was wondering what types of interventions have been tried and what types of research is out there, where you might find some of that research if you're interested in learning more about it.
Well, I think a good place to start, since you're in Eugene, is go and find Phil Fisher, who's at OSLC. He is the man who developed the parent training program that we're using. And he developed it for foster parents, because so many foster kids and parents have a difficult time, which results in the child being placed in different homes like every 6 months. So he developed this program, and he showed that it improved relationships, stability of placement, and it dropped cortisol levels. Cortisol is a chemical that increases with stress. So you can measure that in saliva. And his parental training program was exceedingly effective. And so we just adopted it for use with the parents of our Head Start group. So that's where you should go, right to him.
See one more question, all right?
So there are critical ages for many of these things, but some adults could take up a hobby, at the age of 30 or 40, and study something that normally is associated with a critical age, and make improvements in that area and enjoy it, and maybe stave off Alzheimer's and so forth. But they don't have privy to the plasticity that they have at the critical age. So what is the mechanism whereby they get improving the skills at that late age?
Okay, the question is, what are the mechanisms of plasticity in the adult, and how might they differ from the mechanisms in the child? And that's a big field of research that Mike Merznick can talk about a lot too. But as I said, in adults, we change the weights of synapses by use. That's how we learn as adults. So the more a particular connection in the brain is used, the stronger it gets. The less it's used, the weaker it gets. And we know what the NMDA receptors are important in mediating those changes in connection weights in adults, and they are in children too. So many of the mechanisms important in adult plasticity are important in developmental plasticity. But their developmental plasticity, there are more mechanisms, like redundant connectivity that I talked about, and there are chemical differences, and there are more NMDA receptors in young brains. So young brains are different. They share plasticity mechanisms, but they're not the same. This man has a question.
Well, all right, we'll take another question here.
Do we know of other countries or even other states that have done significant work with parents, either prenatally or immediately after birth? Because do we not know that real low SES or very young parents or uneducated parents have a higher incidence of negative utterances to children, geometrically greater than positive utterances, would be such an easy thing to teach and correct. Do we know of other states or places, countries that have done it from birth, trained parents, on these few simple issues?
Yeah, the question is, what programs are in place across the country, in different states, in different countries? Oh, this is really a burgeoning field right now. I mean, in Canada, for example, there's a whole federal initiative to get this kind of information, to parents across the country. They're putting lots of money into it. They've got scientists on board. It's amazing. Now here, we have, of course, Birth to 3 and Success by 6. And Success by 6 targets low SES groups. They're the ones who put those ads on the TV called Cherish Your Child, for example. I mean, they're just little sound bites, and we're working with them to give them some of this material too, so they can make more little sound bites. But But I don't know if there have been really careful follow-up studies to show the efficacy of what they're doing or to try to even amplify what they're doing. And it's not really my domain of expertise for sure. Do you know anything more about that, Marjorie? Or Jessica?
Well, I think we'll end the discussion. Been a very lively discussion. I think Helen will probably stick around for a few minutes so you could ask her individually. In addition, don't forget that tomorrow morning we'll start at 9 o'clock. We have 2 other world leaders in the field of brain plasticity. They'll talk specifically about educational matters. So I hope you'll join us. I hope you'll join us from Bend, and we'll also have Portland and LeGrand tomorrow. Thank you all very much for coming.